anaphase stage, they become little stumpy, little shortened. And these structures, thick filamentous structures are present where the chromomeres will be attached to it. Whereas in case of the resting phase, that is during the prophase and the interphase, they will be thread-like, coiled, elastic, contractile structures. And when you see the movement of these chromosomes during the cell cycle, you see that they start bifurcating. That is, they will be dividing and then they become V-shaped or J-shaped, or the which is attached to the microfibrils, microtubules, and they are being pulled out. During this process, you will see the shortening or flattening of the chromosomes. So that is how they keep changing their structure during the cell cycle. In prophase and interphase, it is thread-like, and in metaphase, they are flattened filamentous structures, which we call it as fully formed chromosome. And when we study the structure of the chromosome, it is the metaphasic chromosome we will be studying. Based on coiling, whether it is thickly coiled or it is lightly coiled or it is darkly stained or it is lightly stained, we can divide this chromatin into two forms, that is heterochromatin and the euchromatin. Heterochromatin and the euchromatin. Heterochromatin, here the coiling, it is a condensed coiling, that is the coiling of the chromatin. I'll just show you one, I'll share one figure with you. Coiling, what exactly you mean with coiling? This is the coiling of the chromatin. You can see darker coiling and the lighter coiling. The lighter ones are euchromatin and these dark ones are the heterochromatic region. And when you see the chromatin material, it will be either twined very closely or it is loosely twined. Based on that, the structure of this chromatin will be differing. And the heterochromatin, this based on the condensation of the coiling. We say heterochromatin. This heterochromatin, and during transcription, this heterochromatin is inactive and it helps in the manifestation of the chromatin. Manifestation is giving the structure and form to the chromatin, but it is inactive during the transcription phase. And this heterochromatin is seen in the chromomeres, then chromocenters and the knobs that are there. Generally, these knobs will be seen in plant material. And here we take satellite DNA as the knobs of the chromosome. Coming to euchromatin. Euchromatin, the coiling is less. It is active for transcription. When the coiling is less, then the genetic code, the genetic material, it is easily available for the transcription purpose. So therefore, it is active. When coiling is more, transcription will be low. But that is because they are highly packed together. Whereas when it is loosely packed, then you will see that the transcription can occur. So during transmission, what exactly happens? That is, mRNA will be transcribed from the rRNA on the DNA to form the respective protein. So this is a process that will be occurring from the chromatin material. So euchromatin and heterochromatin. What is the significance of this heterochromatin? Again, heterochromatin is of two types, facultative and the obligative chromatin. When you talk about the facultative chromatin, this facultative chromatin, heterochromatin, it occurs during the cell cycle. And this the euchromatin, it is it shows its variability. That is, what do you mean by variability? That is, it can be converted into heterochromatin anytime or back to euchromatin. That is why we call it as a facultative heterochromatin. It is interchangeable. That means during the cell cycle, this heterochromatin can change into euchromatin, loosely coiled, and then it can become a coiled structure. So then, where exactly you find this facultative? You will see that this facultative chromatin, you will be seeing it in the sex chromosome, the bar body that is present, bar body in human female. Generally, this is called as the drumstick chromosome, which is present in the female. The X chromosome, it is having heterochromatin. This helps in the identification of the sex. That is, when you take the cheek cells, that is from the cheek, if you remove the cells and then observe for the chromosomes, you will see the bar bodies in female. If that Barr body is present, you can say that it is a female or it is a male person. So identification of the sex, it is Barr bodies will not be present. And then coming to obligatory, this is present in the cells where it is permanent, constant, its position all along the length of the nucleus. It will not change. If it is heterochromatin, it remains heterochromatin only, especially in case of centromeres and all that, it remains as a heterochromatin only. Euchromatin, this is active, as I told you, euchromatin is transcriptally active, and it is having a lot of advantage over the heterochromatin because where the proteins can be synthesized or transcription can occur. During embryology, differentiation occurs only in the euchromatic cells. Then coming to the fine structure of the chromosome. Before going into the fine structure of the chromosome, let us study certain aspects of the chromosome. Like what type of chromosomes do we come across, or what is a chromosome number? What is the shape of the chromosome? Then what is the size of the chromosome? When you see the size of the chromosome, this I think I'll come into the next part of it. In the fine chromosome, we will, as I already told you, the chromosomes, they will be changing their shape and structure during the different phases of the life. Is nothing but the chromatin material, and it appears only during the cell division. And one more term that is called as a chromatid. Chromatid is nothing but it is a part of the DNA molecule. When it is attached with the centromere, or if the centromere is present in the structure, the two arms of it will contain the coiled structure, which is called as the chromonema. So that will come across a little later. In prophase, again, you will see that the chromosome is full, whereas in metaphase, the chromosomes, it has two chromatids because it will be joined with the centromere. Whereas in case of a prophase, you will not be able to see the centromere. In case of metaphase and present, whereas in anaphase, there will be only one chromatid because it will be totally driven towards during the cell cycle. Coming to the chromosome number, and this chromosome number is constant for all the type of the species. If you say take any type of a species, it is constant. Like for example, in plants, in the like yeast, it will be having 8 chromosomes, or in pea plant, as you have already seen, that it will have 7 chromosomes. Like that, it is a determined set of chromosomes will be present, and this is having a phylogenetic and the taxonomic significance. The number is giving us the phylogenetic significance as well as the taxonomic significance. Now, based on the number, we can say that there are different types of the terminology that is being used. That is euploidy and euploidy. Euploidy is full set of chromosomes. Full set means whatever number of chromosomes one particular cell should have, that the number of chromosomes should be present, and that phenomena is called as a euploidy. It consists of full set of the chromosome. Like this can be again based on the number of chromosome that it has. Generally, we see that one particular cell, that is the gametic cell, will have haploid number of chromosomes, whereas the somatic cells will have a diploid number of chromosomes. So haploid number, that is represented by N, diploid by 2N, triploid by 3N, tetraploid by 4N, and polyploid. When you talk about the polyploid type of the chromosome, this polyploid, it is the number which keeps on increasing from one cell to the other cell, and from where the number of the basic number, you call it as a basic number of the chromosomes. From there, 7 chromosomes are there. 7 will later on will become 7 plus 7, 14, then 14 to the next 28, like that. When the polyploidy is, the number of chromosomes are increasing or duplication is taking place, the basic structure will be considered as the base number. And the full set of the chromosomes that are present in a gamete, it is known as a gametic chromosome. And that particular composition a particular cell. Chromosomes that are present in a particular gamete will give you the genome of that particular individual or particular cell. And generally, when you see the sex cells, they are always haploid, whereas somatic cells are diploid. Now, in case of human beings, when you talk about the diploid type of cell, in human beings, 44 chromosomes are diploid. And all these chromosomes are considered to be homologous. Homo means similar. Homologous. They will be always be in pairs. So homologous pairs of chromosomes will be present. They are 44 in number. Whereas the two chromosomes that are there, that is called as a sex chromosome, these sex chromosomes will differ in male as well as in female. In case of female, it is X and X. Therefore, it is called as homomorphic. And in case of male, you call it as heteromorphic because one is X. 44 diploid set of homologous chromosomes are called as autosomes because they are present in the somatic cell and the sex cells are present in the gametic cell. Coming to aneuploidy. Aneuploidy, where you will not see any change in the chromosome number except that there will be a loss of or addition of any of the chromosome. The sex will be same, but there will be addition or there will be deletion of a chromosome if one chromosome if one chromosome is been added, then it is trisomic. If two chromosomes have been removed, a pair of chromosomes are removed, then it is called as nullisomic. So here, in case of, as I told you, the human beings, 46 chromosomes, you have homologous as well as the heteromorphic or homomorphic type of chromosome. Next terminology type. Karyotype, it is a total description of the nurse with reference to the size, number, detailed structure of the chromosome, and which helps in the identification of the set of chromosomes which is specific to each species. That phenomena is called as karyotype. The karyotype is nothing but it is described as the concept, or it is described as a step wherein the chromosomes can be differentiated for the identification of a specific type of a species based on certain attributes of the chromosomes like size, number, structure, etc. Ideogram is nothing but the diagrammatic representation of the entire chromosome set. So ideogram is nothing but it is a diagrammatic representation. I think in your practicals you have done the chromosomes and you have arranged them. So that cell arrangement it is called as ideogram. Now coming to the different arms of the or shape of the chromosomes. Before going into shape of the chromosomes, let us go to the size of the chromosome. The size of the chromosome ranges from 3 micrometers. Micrometers we can't even see with our own eyes. 3 micrometers in fruit fly. Fruit fly nothing but the Drosophila. And in man, it is 5 micrometers. So you can just imagine what will be the size of the chromosome. That's the reason why when you see a mitosis slide or a meiosis slide, you have to magnify 100 times to see that particular chromosome. And one more point we should know that I said 5 micrometers is the size in man, but it is not the same with all the chromosomes. That this is an average of the chromosome. If the chromosome length is increasing or decreasing and all that, we'll have again a type of genetic problems that are there. But here, when we talk about this, it is just an average of the cell size. Generally, in a particular cell, when you see the 46 chromosome, some of them are like small, some of them are large or something like that, isn't it? The size will be differing. But on an average, we consider it to be as 5 micrometers. The largest chromosomes we come across is the Lambrish chromosome in the vertebrate oocyte. In oocytes, we have two different types of chromosomes, that is Lambrish chromosomes, and also in case of insects, we see polytene chromosomes. These are the modifications, like they are separate different type of the chromosomes, Lambrish chromosomes and polytene chromosomes, where the size is even more larger than what is expected in the normal cell. Coming to the shape of the cell. When you talk about the shape, it depends upon the arm ratio. What is the arm ratio? This is a changeable fragment, that is the two arms of the chromosome which is attached with the centromere keeps changing. The position of the centromere will bring about the difference in the arm ratio. So therefore, here you see that in case of the number, you see that the shape of or the position of the centromere keeps changing. When the position is in the center, when the centromere is in the center, both the arms of the chromosome will be same. Both the arms of the chromosome will be same, and it will acquire a V shape. And generally, it is considered to be the metacentric chromosome. And this you will be seeing during the anaphase. And also examples, we can say that most of the amphibians, the metachromatic chromosomes will be present. Then coming to submetacentric. Submetacentric is not in the center, but not at the terminal end, just above or near to the center, but not at the center, slightly towards one side, and therefore it is shaped as L or tick shape. Therefore, this type of chromosome, it is called a submetacentric chromosome. Then acronymatic, this is present towards one end near the pole, not towards the pole, not at the pole, but near the pole, making one short arm and one long arm. Especially you see it in locust. Then telocentric, the centromere is in the terminal end. And in most of us, we know that telocentric chromosome is not present in human beings because it gives loss of one arm. It is a mutation that occurs, isn't it? There's a completely a loss of one arm. So apart of, because the chromatin material or the DNA which comprises of the required set of the genes will be lost completely, and therefore be a genetic mutations that can occur. Therefore, this type is very rarely found, or it is not at all present in case of human beings. Eccentric, centromere is totally lost. No, not moving to, I mean, it is not towards the poles, and therefore what happens when the centromere is not there, those chromosomes, they become useless because they cannot bind or combine to the spindle fiber or the microtubules during the cell division. Therefore, when they cannot uh, take part in the cell division, they will be completely lost from the next generation. So they will be lost in the cytoplasm. So eccentric chromosomes, they do not have a centromere, and thus they are totally lost in the cytoplasm because the centromere is the major portion from where it will be attaching to the microtubules during the movement of the chromosomes in the cell cycle. Okay, are you all understanding?
Yes, ma'am.
Are you all understanding? Yeah, okay. My pace is slow or it is fast? Okay, speed is okay?
Yes, ma'am.
Okay. So arm ratio, it is considered that as based on this, we can say that arm ratio, what exactly arm ratio is? The relation of one arm to the other. It is a ratio of one arm to the other. This is most important factor which will give us the type of the chromosome, what a particular species is bearing. And it remains constant for a particular type of the chromosome. For every cell, this remains constant. And based on this only, we will be carrying out the karyotyping. Is it clear? Now, coming to the detailed structure of metaphasic chromosome. When we talk about the detailed structure of the metaphasic chromosome, we should know about certain things like what is chromatid, what is chromonema, what is centromere, what is primary constriction, what is secondary of the chromosome. You can see this. This is a chromosome. This is a telomere. The end portion where there will be repetitive DNA will be present at this particular end, and there will be a small constriction, and this constriction is called as a secondary constriction. Whereas near the centromere, you will find the first constriction, secondary constriction 1 and secondary constriction 2. And where the first or the primary constriction is there, at that point, you will be finding the centromeres. And associated with the centromere, there will be a plate-like structure, which you call it as kinetophore. And that to the microtubules. So when you see chromosome, you see that inner two coiled structures are present. And this coiled structures, the thread-like structures, it is the chromatin. When the chromatin is coiled, you call it as chromonema. So the chromatin or chromonema, it is generally a equal type of names, or it is a synonym that this can be used. Either you can say chromatin material, or you can call it as a chromonema. Whereas the chromatid means this part is one chromatid, and below the chromosome it is one chromatid. So chromatids two, whereas chromonema, it is the internal coiled DNA. segment or the chromatin material. And the chromosome consists of the matrix of the nucleoplasm, and it is surrounded by a pellicle, whereas chromonema or the chromatin is not surrounded by a pellicle. And below the secondary constriction, you will find the satellite DNA. Most of the chromosomes do not bear a satellite chromosome. Very few will be bearing the satellite chromosome. And the secondary constriction is mainly for the nuclear organizer, that is for the synthesis of that nucleolus, which contains a rich amount for the transcription purpose. So this is just a brief structure of it. Let us go to each one of this. What is chromatin? Now, chromosome is present inside the matrix without a pellicle. And then chromatin, it comprises of DNA molecule in metaphase, and two chromatids are present in anaphase. I told you that during this particular phase, you will be finding that it will be broken down because of the kinetochore that is there. It will be giving two arms of the chromosome. That is why we say that two chromatids are present at the metaphase, and one chromatid at the anaphase, because it has already been removed or to two sides from the metaphasic stage where the chromosomes are like this, where you have the kinetochore and two arms of the chromosomes will be there. And once it goes towards the anaphasic stage, the chromatids are being broken down. So single type of chromatids will be present. Now, chromonema. Chromonema, it is nothing, it is not chromatin or the DNA molecule. During interphase, this chromatin is present like thread-like manner, and the nuclear membrane is called as a chromonema. It is early in the earlier stages of the formation of this chromatin, the condensation of the chromatin, you call it as chromonema. So this is nothing but it is a condensation or a condensed chromatin material, which call it as chromonema. Coming to centromere. Centromere, it is a special region where chromonema is almost uncoiled, forming a disc-like structure with special proteins, and is meant for the attachment of the chromosome to the microfibrils at the centromere. So centromere is that position where you will see that the chromonema is uncoiled and it produces a plate-like structure, which you call it as a kinetochore or the centromere. This kinetochore will be attaching to the microfibrils. So this centromere formation or the formation of the centromere, it is also known as a primary constriction on the chromosome. The primary constriction is also called as a centromere because here the chromonema is uncoiled and it has a protein cover. Based on the centromere, we have different types of the chromosome, that is metacentric, submetacentric, telocentric, acrocentric, etc. The chromosomes are tightly coiled, dense globular structures with a core protein. This we will be talking little later. It has a protein and a DNA strand together. These proteins are nothing but the histone proteins. And when you see the structure or the size of this chromosome that is being formed, it is 50 angstrom unit. So the globular protein that is there, which you call it as a histone, and the DNA, they will be coiling together. And this model is called as a nucleosome model or the solenoid model. In the subsequent lectures, we will be talking about the solenoid model. The areas which are thick of globular structures, it is called as a chromomere. That is what I told you. In the chromomere is nothing but where you will see that the chromosome here, you can see these two dots here. These are chromomeres wherein it forms a thick plate-like structure, which you call it as a kinetochore. Now, they disappear during anaphase, metaphase, and telophase. Or you will be seeing that having the beaded appearance, whereas in case of as the cell cycle is going on, this chromomeres or the centromeres will be disappearing because they will be pulled towards the sides and the two different cells are being formed, and therefore there is no need of the centromere or the chromomere. The same structure I will show it little here. Say this is the chromosome structure, this is a centromere, and you can see the chromonema which is being coiled, and this is the pellicle of the cytoplasm that is there, and this is the matrix of chromoplasm that is present. And when you see it as the two chromatids, when we talk about, see, this is a centromere, and during the metaphasic chromosome, what is happening? The same chromosome, it has been divided into two. One chromatid, two chromatid. Did you understand? So one chromatid. And in internal structure, you will see that this is nothing but a chromonema. And here you can see that it is much coiled in the chromatin part, whereas in the centromere part, it is not coiled. Okay, so then the same thing in another one with all the labelings what we see, the repetitive DNA. You will not see the coiling is not there, but repetitive GC component will be more. Chromonema, generally the coiling of chromonema, this is a structure, it will be coiled like this. But diagrammatically, we represent this coiling as small bulbs that are being formed. These are called as chromomeres. The small bulbs that are being formed, they are called as the chromomeres. So chromonema, it will form into small bulb-like structures of the coiling. And what do you mean by intercalary heterochromatin? That means in between. It is present in between two euchromatin. So it is intercalary heterochromatin. And this is the first constriction what you see, that is called the secondary constriction 1, because secondary constriction 1 will be present towards the satellite DNA. This is a satellite DNA. And where the nucleolus is being formed, that is nucleolar organizer is there, that type of constriction is called as a secondary constriction 1. So the other constriction what you find towards the telomere, and most of the part you will find that it is the heterochromatin and the euchromatin which is there. Euchromatin, as we said, that it is the active part for the transcription, where the heterochromatin remains darkly stained and inactive for transcription. And this is a primary constriction. Within the primary constriction, you will see that the formation of thick platen structures, which you call it as a kinetochore. And then coiling of the DNA. If it is closely coiled like this, in this you are seeing, it is closely coiled like this. When it is completely coiled, you will call it as paranematic coiling. When there is a distance or loosely coiled structures are there, then it is called as a plectonematic coiling. These are the structures of the chromosomes, what you see. This is a metacentric, both the arms are same, submetacentric, not towards the center, but not towards the pole, whereas here it is towards the pole and telomeric at the end, and this is the dicentric. So, sometimes, in some cases, you will find that instead of one chromocenter, now you will kinetochore, you also find that there are multiple of the kinetochores present throughout the length of the chromosome. So here, one example what we have taken is a dicentric chromosome, where we have the two centromeres are present. So it's a very rare foundation or very rare case. Now, what are the functions of the chromosome? When you talk about this chromatin or the chromosome that is there, so it is a protein which is condensed, the chromonema or the chromatin, it is a protein which is condensed with DNA, and DNA is already coiled DNA, and it is clubbed with the protein histones. And this coiling, it makes it inactive. When it uncoils, becomes active. Chromosome helps in the identification of the chromosomes of a particular species. So she is born, like what type of chromosomes that are there, which are predominant, or if there is a loss of any type of chromatin material, etc., can be identified with the help of a chromomere. And this chromomere can be used by using the karyotyping or banding of the chromosome, so that way the chromomeres are present. And most chromosomes has one centromere, and in some cases you will find that they are all along the length of the chromosome. And when it is having all along the length, it is called as a holocentric chromosome. And here it will be attached to the microfibers at many centers, dicentric, polycentric, etc. Whereas when it has only one, it is called as a monocentric type of the chromosome. Acentric, during division, centromere is carried by one of the chromatid. So other will not have any type of the chromomere. So that is how we get the acentric type of chromosomes. So acentric is that during the division, it is not equally divided, and that chromocenter or the chromomere is not being divided equally between the two chromatids. So one of it will be taking away and the other does not have it. That is how we will be having the acentric chromosome, and it will be lost in the cytoplasm. Coming to the secondary constriction. Secondary constriction, it is a specific in location. It is very, very specific. And here you will see that the matrix is reduced. They are not present in all the chromosomes. Secondary construction is not present in all the chromosomes. In humans, only specific chromosomes having secondary constriction is 1, 11, 13, 16, 21, and Y chromosome. With this, we will be able to identify whether they have a satellite DNA or not. Secondary constriction 1, the region of secondary constriction, it is actively participating in the synthesis of 18 sRNA, 28 sRNA, and the transcription process. So therefore, it is responsible, or it is responsible for the production of nucleolus. So this secondary constriction one that is there, it disappears during the formation of the nucleolus, or this secondary constriction that is there, it is helpful in the transcription process. And once that is being done, after division is being done, again it will reappear. And these are also not present in all the chromosomes. That is, as I told you, that these constrictions will not be present in all the chromosomes. Similarly, only in 13, 14, 15, 21, and 22 chromosome, you will be finding this secondary constriction 1. The region below the secondary constriction is a rounded structure called as a satellite DNA. This satellite DNA, it is also called as SAT DNA. Now, coming to the terminal end of the chromosome is called as a telomere. What is the significance of a telomere? Like when we talk about this telomere will allow the chromosome not to stick with any other chromosome, and it will be attached to the nuclear membrane. It permits the function of the two ends of the same chromosome, and it will also help in the attachment of the chromosome to the nuclear membrane. And when telomere is there, it will not allow the attachment of other chromosome part or other material to attach to it. When these are lost, then you will be seeing that there will be mutations occurring because of the stickiness of the chromosomes to one another and leading to the mutation condition. So they are the terminal ends which will be attached to the nuclear membrane, and they will not allow the fusion of two different types of the chromosomes. This has already been done. The heterochromatin is found in telomere, primary constrict. I have given it to you. Now, coming to so many theories have been put forth. So two theories which basically are being taken. Dewdraw's model, that is folded fiber model. According to this theory, what they say is the fibril, that is the chromonema that is present, that the DNA fragment that is there, it is of only 20 angstrom units. When it combines with a protein, it becomes 70A, that is 70 angstrom unit size. That is, it increases its size. So here, the coiling will be, single coiling will not be efficient. So carry large amount of information. It cannot be a straight structure. And when it is a straight structure, the chromosome structure should also be quite large. So therefore, this chromatin material, it will be folded, coiled. So how is it coiling? That this theory is showing that when, that is Chris et al. In 1964 said that this molecule, it will combine with the protein to form 70 angstrom unit nucleoprotein, which in the sorry, I have written it as absence here. It is the presence of calcium ion. It will be transformed into a fiber, which is 100 angstrom units. And this 100 angstrom unit, it is a large molecule. It cannot be a straight molecule as such. So therefore, it has to coil. Like for example, when you are taking the long thread, and when you are putting it down, that long thread will be coiling and then it will stay. Whereas if you're taking a small thread, if you throw it down, it will be a straight molecule only. So similarly, here also, as it is a very long thread, you will see that it gets coiling. And this coiling will be because of the attachment between the base pairs that are present. And later on, the folding, in order to increase the complete folding structure, here you will see that it will convert into the further folding, which we call it as a B fiber. B fiber will be of 200 angstrom units. Like that, the model has been proposed based on the number of base pairs that are there, based on the length of the nuclear material, the coil coiled. Therefore, it will attach with the protein in the presence of the calcium ion. It will gain the strength, that is 200 angstrom units, that is weight. 200 angstrom unit is nothing but the weight. 20 angstrom unit is the weight. 70 angstrom unit is the weight. So as the weight increases, there the coiling will increase. According to the Dewdrop model, he says that it is the folded fiber. It is same as it is when it is a single molecule of DNA. The molecule of RNA, it undergoes vertical and horizontal loop. It will, because it is a single thread and it has to condense in a particular material, so it will undergo vertical and horizontal folds. Because of these horizontal folds and all that, we will be finding that Lamprecht chromosomes will be seen, that where it will be giving the puffs of the chromosome. So here, according to him, he says that it is the that is chromosome that is there. This single stranded chromatin that is there, this single stranded chromatin, as it has to condense a lot, it will undergo vertical and horizontal loops. Whereas according to this model, he says that uninem, that is a single model, what he says that here the protein will combine, calcium will combine, give the weight to it, and therefore the coiling can occur. Here it says that vertical coiling as well as the normal coiling will be occurring in case of the chromoneme. That is about different thing. Now coming to the last part of the chromosome structure, it is the molecular structure of the chromosome. When you talk about the molecular structure of the chromosome, the what we have seen till now is the chromosome. It is a nuclear material which is helpful for hereditary mutations, etc., etc. You've seen what are the parts of it. It will have a chromatin, it will have a chromoneme, and then it will have constriction, primary constriction, and the secondary constriction. Primary constriction is present associated with the centromere, and then the secondary constriction 1 is associated with the satellite DNA, secondary constriction 2, nuclear organizer, and this is what we have studied. This chromosome is being formed when you see the molecular structure, and as the uninem model was telling, that the fiber that is there, it will be attached to the protein. The protein will coil together and form the DNA molecule. So to know that model, we Korenberg and Thomas, they proposed this particular theory, and they said that this chromosome that is present, it is made up of certain chemicals. What are they? One is DNA, the second one is the proteins, the third one is non-proteins, and that is proteins are histones, non-proteins are other fibrous material. So histones and non-histones majorly constitutes with the DNA and histone. Here you see that how the molecule is being folded. To know that, I have put them in smaller points. That is, first DNA will interact with the tetramer. Tetramer is 4 molecules of histones. What type of histone? Histone 3 and histone 4. Histone 3, 2 molecules, and histone 4, 2 molecules. 1 minute. Histone 3, 2 molecules, histone 4, 2 molecules. It will form a tetramer. To this tetramer, DNA will be coiling around it. And to this 4 structure, 2 molecules of H is another histone protein which is having segment A and segment B, or subunit A and subunit B. So it will combine with the oligomers. That is, oligomers in a sense that they are the carbohydrate content, non-histone protein. So they will be combining with the complete tetrad structure along with the DNA. And this again, they will be as combining with the proteins H3, H4, H2A, H2B, along with 200 base pairs of DNA. So DNA will be coiling around this. So it will be just like your Sutli bomb. You have a Sutli ball, and on that, the Sutli has been coiled. So DNA will be coiled on this histone protein. So it will be coiling on it, and then it will make a repetitive protein. And repetitive unit, the DNA will be coiling on it repetitively, and that's how it will form one particular. There will be a H1 molecule which is covering the entire molecule, and therefore this one segment will be continuously repeating. So tetramer makes a core unit, oligomer will be determining the spacing and giving the flexibility to the structure. So this I will show you here. You can see that tetramer formation, On this tetramer, you will have a DNA that is there. And this DNA, it is coiling, as we said. Another tetramer, coiling. H1 proteins will be helpful in joining. When you see the puffs or that what you are seeing the of the coiling like this, and it will give a beaded appearance to the complete nucleosome, or it will be giving a beaded appearance to the complete chromosome. Do you understand? So that is the molecular structures. When you see the molecular structure of the chromatin material, you will see that it is made up of DNA and the histones, which are coiled together to form a solenoid structure of a chromosome. Similarly, in case of telomere, you will be seeing that it is having a repeated structures of G and C. What is G? Guanine. C is cytosine. So repetitive structure of this base pairs, you will be seeing in kinetochores, you will see that at the region of the constriction, they are in the constriction region, as I told you that they are joined together. And this at the constriction, they are joined, but they are not coiled. They are not coiled. So two chromatids are joined together. They will be very closely attached together, and the coiling is less. And this is a site for binding with the microtubules. So this is about the structure of chromosome. Is it clear? Clear?
Yes, ma'am.
About the structure of a chromosome. You need to describe that what is a chromosome, how many chromosomes are present in a particular individual, what is that set of chromosomes called as, that is chromosome number, euploidy, diploid, haploid, etc. And then you have to give the structure of the chromosome, that it made up of the kinetochore, the arms. Based on this centromere, you will have different types of the shapes of the chromosome. And this chromatid consists of the chromonema. Chromonema is nothing but the chromatin. This chromatin is highly coiled. And why is it coiled? This is because of the DNA and the histone proteins, which are coiling and giving a beaded appearance of this chromonema. And then you have to talk about this chromatin. It is based on the basic stains. It is darkly stained or lightly stained, that is heterochromatin, and the euchromatin. And this heterochromatin is facultative or obligative. Why? Because the transcription has to go on. Euchromatin is basically useful for the transcription, whereas heterochromatin, there are two types, facultative and obligatory type. Facultative will be continuously changing either to U to heterochromatin depending upon the situation, whereas euchromatin will not, obligatory chromatin will not change. It remains same. So based on the centromere or the primary constriction, we have different shapes of the chromosomes that are there, metacentric, etc., etc. And we know that these centromere will be one in one chromosome, but at times you also find that there are number of centromeres present, which is called as a holocentric type of the chromosome. And this is what you are supposed to talk about it. And this is the models of the folding. It is just an added information to you. Coming to what is a karyotype. Now, karyotype, as already you know, that it is nothing but it is the arrangement or knowing the complete set of the chromosome. So we say that it is every group of individual, they comprise of a set of chromosomes which has certain features, certain attributes. What are they? The number is constant, the size is constant, the shape of a particular chromosome will be constant. So these structures, when we study individually in comparison to each other, then we'll come to know that what is the difference between one species to the other species. So knowing the chromosomes and then identifying that chromosome and relating it with the primitive one, similarities, dissimilarities we have from each other. Based on these, we will be able to identify the species, the level of the species, or the taxonomic of the species. Therefore, karyotyping is very, very important, and it is based on the attributes of the chromosome, that is the constant features like number, size, shape of an individual. The term karyotype is given to a group of characteristics that which identify a particular chromosome set, and is represented by ideogram, where chromosomes of haploid set of organisms are ordered in a series of decrease or arranged in a series of decreasing size. So this already we did it in our practicals, I suppose, that the karyotype, it comprises of the complete set of the chromosome. And when you arrange this complete set of arrangement, this complete set of arrangement particular order now from the higher size or larger size to a smaller size. Now, coming to what does this karyotyping suggest, or what is the use of this karyotyping? When you see karyotype suggests the primitive or the advanced features of that particular organism. So based on the bands, based on the euchromatic features, based on these banding you have found out, based on the type of the chromomeres or centromeres that is there, you will be able to identify what are the primitive characters, what are the advanced characters of that particular species. It shows a large difference between the smallest and the largest chromosome of a set, and having fewer metacentric chromosomes is called as a asymmetric karyotyping. Now, this point I would like to express it, that it also gives us these condition where we are able to identify the smallest and the largest chromosome and find. And here generally when we see, because when we're talking about the chromosome, we generally take during the metaphasic chromosomes only, because we have the double set of chromatids to study about it. So therefore, during the metacentric chromosome, depending upon how many types of the metacentric chromosomes are there, if you have fewer number of chromosomes, this is called as the asymmetric karyotyping, and it has a very good advantage over the symmetric type of the karyotyping. In symmetric karyotyping, what we see is that the set consists of the regular or the normal type of the metacentric chromosome. So based on that, the difference, if we are finding any type of or lesser number of metacentric chromosomes, we'll come to know that there is drastic change over the normal set of chromosome. And this karyotyping is brought about by banding of the chromosome. And the banding is based on the GC and AT, that is purine bases that are present, purine pyrimidine bases that are present on the DNA molecule. So which particular region is rich with GC, that is guanine, cytosine, or adenine and thymine, which bases are, which regions is having highly repetitively concentrated GC or AT. Based on this, we can identify different types of chromosome. I think this karyotyping already you have done it. So this darker shade. So what type of banding we come across here? Which is this? If it is a G banding, or it is a R banding, or it is a N banding, depending upon the type of the banding technique, we'll come to know that what is the nature of the chromosome. Now, based on this GCAT staining, we have different types of binding. They are Q binding and G binding. Q binding. Here in Q binding, we will be using a stain, which is a fluorescent dye, which is called as quinacrine mustard. It is being used, quinacrine mustard. This is the stain, fluorescent dye that is being used. So this will go and attach to the components, AT component or GC component, and it will give the bands. When it gives the bands, it will be fluorescent, and based on the number of banding, we will become knowing that which chromosome is that, and is there any deformity in that chromosome, or if it is rich in particular component. So based on that, we'll be doing this is called as a Q banding. And what we are doing here, this is nothing but the Q banding only. Coming to C banding, the next type of banding, that is G banding. G banding is we are staining the complete chromosome set with a GIMSA stain. Both Q and G bands correlate with the chromomeres, where the chromomeres are present. So it will be correlating with that. And this Q banding and G banding can be observed during the leptotene and pachytene stages during meiosis, because chromomeres, how many chromomeres will be there? As I told you that the binding or the coiling of the chromosome that is taking place, that is, we are calling it as chromomeres. Wherever there is a loop forming, the constrictions are there, we will be able to see this chromomeres. So depending upon the number of chromosomes, if Q banding, G banding is being done, you will be seeing that the chromomeres are increased in number. That means that particular stage is in a leptotene or a pachytene stage where it is a beaded appearance. C banding is, it is pre-treatment of the chromosome with the procedures that will denature the DNA. Denature the DNA in a sense that, and before doing the C banding, what we need to do is we have to uncoil the DNA. That is called as denaturing of the DNA. So this uncoiling of the DNA can be done by treating it with certain chemicals or certain stains like Giemsa staining, etc. And then restain it with another type of a stain, which will be staining the chromatin material. That is, here, once you are denaturing the DNA, again you are staining it with instant staining in kinetochore region, which will localize the constitutive DNA. Constitutive is obligatory DNA. So when you localize this DNA, you will be able to identify what type of constrictions you are able to see. That is, the secondary constrictions can be identified by doing the C banding. So the secondary constriction of which chromosomes? 1, 9, 16 chromosomes, and the distal segment of Y chromosome can be stained by using the C banding. This method provides the polymorphism because C bands may vary from person to person. Why? As I told you, the secondary constriction may be present in many or may not be present in many. So based on that, we'll be able to identify the polymorphism that is present in different type of the chromosome sets, that is in different species, because of the different amount of satellite DNA in the chromosome. And when the secondary constriction is there, I told you that there will be a satellite DNA. Satellite DNA may be present, may not be present, and if they are present, what type of satellite DNAs are present can be identified by using the C banding. Similarly, R banding, it is a reverse of Q and G banding. That is, when you are using these two dyes, you are getting darker bands. R banding is, it will give you will study the lighter bands. That is the reverse banding of Q and G. Then T banding, this is when you want to identify only the telomeres, whether they are present in the chromosome set or not. You use a T banding. It will go and stain only the telomeric region. F banding. F banding is generally we use this particular stain, that is the fusion stain, which will be giving the banding. And this banding, again, we have labeled it as P and Q. We'll come to it little later. N banding. N banding is only when you want to find out the nuclear organizer region, when you want to find out where this is located, that is where is the secondary constriction is located. To know that, we do the N banding. And based on these bands, we will be able to identify, they have given it as P and Q arm. P for the short arm, Q for a long arm. Suppose centromere is present at the submetacentric region. There is a short arm and the large arm. So where is the band present? So where is the band present? This is the P arm, this is the Q arm. So if you want to label this particular, how we will be labeling it to the P and bands in long arm and the short arm. Like for example, if we label it as 6P23, when you are labeling it as, suppose we want to label it as like this, that means it is a chromosome 6, and P means smaller arm. Which band you are talking about? It is a third band. Where is it present? In region 2. So this is how we will be labeling the chromosome, and then we will be able to identify, are there any changes occurring in that particular region. Like that, based on this, we can give numbering to each and every set of the chromosome and then find out, compare it with the normal set, and then find out whether there is any changes that are occurring in this particular chromosome bands. Based on that, we can do the further analysis of mutations or heredity or anything like that. So how this banding is helping? Banding is helping us in identifying the reciprocal translocation. That means, is there any kind of a translocation of the material? Like, what do you mean by translocation? Now, see, for example, if we take this particular chromosome and see the banding, see this particular chromosome, see the banding. Fourth chromosome should have the banding like this. And the fifth chromosome should have the banding like this in a short arm. But suppose with this banding, if this chromosome part has been translocated to this particular part, and this has been because we know that during the meiosis, the chiasmata formation during meiosis, the dikinesis stage, there is an exchange of the chromosome material, isn't it? So during that process, are there any translocations occurring? To know that translocations, we will be using this banding technique. And it also helps in the detection of more than 30 chromosomal syndrome, like trisomy, nullisomy, etc. If there are any extra chromosomes, we'll be able to identify what type of, or if there is a deletion of any chromosome, what type of syndrome it is, like Klinefelter's, Down syndrome, Turner syndrome, etc., can be identified by using these karyotyping structures. So what is karyotyping? Karyotyping is the complete study of the complete set of the chromosomes based on the number, size, shape, individual, and arranging them in a particular sequence in an order. And by using a particular different binding techniques, we'll be able to identify the particular regions like nuclear organizing regions, or it is a telomeric region, or it is the secondary constrictions that are there, or it is the P and Q band, etc. So based on that, we will be able to identify. Human chromosomes has been identified, the karyotyping of the human chromosome. This is an example of it, wherein it has been divided into like sorry, 7 type of groups or 7 groups, in which each group has a different set of chromosomes. That is A group, it is first, second, third chromosome, which are large chromosomes. To 4, 5 metacentric, 4 and 5 they are submetacentric. They are not in the center, but they are like then coming to 6 to 12 and X chromosome, they are medium size and submetacentric type of chromosomes will be the centromeres will be seen. 13 to 15 they are large acrocentric with satellite DNA. See, you can see here acrocentric, a little short arm will be there, and there is a satellite DNA or a satellite present on it. Then coming to the next one, 16 to 18. Number 16 is metacentric. Number 17, 18, they are small but submetacentric. Similarly, F the group, it comprises of 19 and 20. They are small and metacentric. You can see 19 and 20. It is small and metacentric. Then 21 to 22, you will see that they are short, acrocentric with a satellite, whereas Y chromosome is without a satellite. Okay, Y chromosome is without a satellite. They are all acrocentric. So this is the complete karyotype of a human being, or wherein you will be able to identify the different set of chromosomes that are there. So karyotyping is useful in not only identifying what it is, but also it is helpful in knowing different type of diseases or mutations that have occurred in the chromosome set. Is it clear? Yes?
Yes, ma'am.
So sorry, I could not present you the typed PPT. This was the notes what I have prepared. And then I don't know how notes will be going to you all. You have the material? You have the material, or you need this material?
Need this material, ma'am. You can send it in the general group, ma'am.
Okay. Where should I share this material? Shall I
In
give
the
it to
general
Kishore,
group,
sir?
ma'am.
Shall I give it to Kishore, sir?
Yes, ma'am.
Head of the
Yes,
department,
ma'am.
he will share it?
Okay, ma'am.
Yeah.
Yes, ma'am. Yes.
Any
I'll
one
let you
of
know
the
for-
student please send
Yes,
the
ma'am.
thing, ma'am. Any one of the
You can send it to
student.
Kishore, ma'am.
Hmm.
Students means again, if the
Better to-
student doesn't share, it becomes.
Yes,
yeah.
ma'am. Yes, ma'am.
I will-
Yes, ma'am.
I'll share the material. Just before your exams, I will be sharing all the PPTs that are there. If the PPT is in a printed format, then and there I will share it with you all. But if it is in written form, I need to prepare it in a PPT. Or is it understandable?
Yes, ma'am. Yes, clear, ma'am.
Yes, ma'am. Yes, ma'am. The class was very
Yes,
good.
ma'am. Yes.
Yeah, if it is clear, then this is more easy for me rather than the
Yes,
printed
ma'am.
ones.
Yes, that's why we want this, ma'am.
Okay, I'll be sharing it with sir. And some of the few points what I've added extra, no, those points are also I'll be able to. I'll just incorporate in this and then give it.
If possible, to send one of the student also, ma'am.
Student. See, I'm totally
We
new
will share.
to your group. I need
Yes,
to get accustomed to one of the students having mobile number or WhatsApp number to that particular student. Then only I'll be able to do it. Maybe another 1 or 2 classes I'll get acquainted. Then we will be able to do it. So what about your attendance? How many of you are attending? You are taking a picture of it?
No, ma'am, there is no attendance.
Today 18 members, ma'am, but
18 members. Can we take a screenshot
now
of the
only 14 are there, ma'am. Now 14 only.
14? Can any one of you record the attendance and then send it to my WhatsApp? Daily, for whatever class I'm taking. You can take a screenshot of it, like how many members they are attending. Take the screenshot of it, and that day's attendance, you send it to my WhatsApp.
Okay, ma'am. I'll share, ma'am.
Radhika.
Yes, ma'am.
Yeah. So today's you take it, and those 4 members who were there, next class you ask them and then put it. My number, and you make a note, no?
Okay.
Nine-
Okay, ma'am.
934
I'll share it, ma'am.
934 888
Ma'am, 1 minute. Tell me, ma'am.
934
Ma'am.
888
Okay.
4299.
4299.
Yeah.
Okay, ma'am.
In this topic, if anybody is having any doubts or something like that, you can just call me back, and tomorrow I will take up another topic and try to complete 2 topics tomorrow. Because
Okay,
it was
ma'am.
a first class for me. I don't know what kind of class I have. I don't know. That's why I've taken a small topic.
Okay.
So tomorrow
ma'am.
I'll continue it. Okay.
Okay. Thank you, ma'am. Thank you so much.
Thank you so much, ma'am.
Sorry for the delay.
No, ma'am.
Okay then. Shall we
Okay,
leave?
ma'am. Okay, ma'am. Thank you, ma'am.
Okay, fine. If anything is there, be in touch with Sir Kishore sir. He will be communicating it to me.
Okay, ma'am.